Hi guys, so I want to take a few minutes here and kind of give you an overview of metabolism. So for some of you, metabolism might be something you've encountered in another class, maybe it's something you remember from your cellular and molecular biology, or certainly it's something you've probably encountered in physiology. So these are actually slides that were from my chemical principles class, so it's kind of instructed at that level, just kind of giving you a bird's eye view, an overview of what we think about when we talk about metabolism. So there's a study guide that I have provided for you guys to kind of go along with this. But again, we're going to move through the rest of the chapters in the course here, assuming that everybody is up to the same kind of point with thinking about metabolism. So again, when we talk about metabolism, what we're really talking about is the chemical reactions that occur in our body that allow us to either break down or build up molecules. And when we're talking about breaking down molecules, we're talking about catabolic processes or catabolism, and anabolic processes or anabolism is kind of the opposite, using energy and smaller molecules to kind of generate the large biological molecules we need in our body. And as we can see, these two often work in concert. This is what we'd refer to as a substrate cycle when we have products from one process feeding into as substrates for another. And so again, it's not uncommon to have fuel molecules that we consume provide the energy and the building blocks for us to build new and different biological molecules that we need. So the metabolic pathways that we're going to talk about, again, are a series of steps in the chemical reactions and biological systems that really allow our body to run. And so one of the things that we'll spend some time focusing on is sort of the central heart of carbohydrate metabolism. So we're going to learn about glycolysis, we're going to learn about the citric acid cycle, and we're going to learn about electron transport and oxidative phosphorylation. And so those three things taken together will really comprise the central heart of metabolism, and specifically the last two, the citric acid cycle and the electron transport chain and oxidative phosphorylation, are things that are used by all fuel molecules. But we'll get into those in a little bit here. So we're going to focus mainly in this lecture here on catabolic processes, so fuel breakdown. So animals and humans, again, get energy from breaking covalent bonds in our main fuels, carbohydrates, fats, and proteins, and we'll talk about the details of each of those in a little bit. And this is an equation that should now be very familiar for you, right? Taking a fuel, oxidizing it, full oxidation of the carbon atoms in the fuel generate carbon dioxide. The electrons that are in this fuel are ultimately delivered to oxygen, reducing it to water. And in the entire process, we try to harness that energy. And again, ATP is how we're going to try to harness that energy. So again, in the body, this energy is ultimately captured in the nucleotide ATP. Remembering that oxidation is the loss of electrons. How does that result in the production of ATP? Well, we're going to learn about how that works in this overview here. So again, remembering that bonds are electrons and electrons are energy, and every time you look at an electrical outlet, remember that that electricity really represents the flow of electrons, that current that allows us to do work. So you have a slide from your textbook that's very similar to this, but this is the one that we're going to use since it parallels the materials that are in this presentation, kind of thinking about an overview of metabolism. So again, here we have our main fuels, and they're found in their polymeric or kind of their biomolecular state. Proteins, again, being a polymer of amino acids, polysaccharides being a polymer of simple sugars, and lipids not necessarily being a polymer, but really the combination of glycerol and fatty acids. Remember a little symbol that we use with our hands to just kind of generate what a triglyceride was. So again, we're going to learn the central heart of the metabolic pathway here. We're going to learn about glycolysis. This arrow here represents the chemistry that's catalyzed by pyruvate dehydrogenase. Then we'll get into the citric acid cycle here, and then the electron transport chain, which is coupled with oxidative phosphorylation. So again, this is our bird's eye view that you might want to have next to you as we kind of walk through these different pathways. So we've mentioned this already as we talked about our different fuel molecules, most of them existing in a polymeric or at least kind of connected state. The only way that we can get these out of our digestive tract is to break them into these smaller units. And we learned about the hydrolysis reactions of proteins, right, with serine proteases, with polysaccharides. Even though lysozyme isn't the enzyme that catalyzes this, we do have lots of glycosylases that facilitate the cleavage of these polysaccharides into monosaccharides, which we can then absorb from our digestive tract. And then lipids, the same thing. Lipids can't get out of our digestive tract unless they're broken down into free fatty acids and glycerol. So again, these hydrolysis products are really what we need to get to in order to absorb things out of our digestive tract and into our bloodstream. So again, big picture, only monomeric forms are going to be absorbed. So big picture here, we're going to kind of go through our bird's eye view before we talk about some details of these processes. Monomers, in terms of monosaccharides or even some of these other ones, are ultimately funneled to pyruvate or acetyl-CoA. These two molecules are what we call branch point metabolites, and that's because there's lots of different fuels that either feed in or out of these substances. So pyruvate, again, being a 3-carbon piece, acetyl-CoA being a 2-carbon piece. Pyruvate can actually be derived by any of the 3 biologic or biopolymer fuels, and the same thing with acetyl-CoA. But we're going to focus on the derivation of pyruvate initially from the glycolytic pathway. Okay. So what we do with pyruvate is generally dictated by whether or not there's oxygen in the room. And so again, here's the structure of pyruvate. That is the product of glycolysis. And again, what we do with pyruvate is determined by whether or not we're operating under aerobic or anaerobic conditions. If, again, there's adequate oxygen in the cells, we're operating under an aerobic environment. And again, there's the need for energy. These 2-carbon metabolites known as these acetyl groups are broken down to carbon dioxide in the citric acid cycle. So again, we're going to get into the details of the citric acid cycle in just a little bit. But again, if we have a need for fuel, once pyruvate is broken down to the 2-carbon fragment acetyl-CoA, acetyl-CoA is committed to the citric acid cycle, and all of those carbon atoms are liberated as CO2. So again, that's one of the places that we get the majority of the CO2 from our fuels. And then we do generate a little bit of ATP, and as we'll see in a little bit, we also save some of that energy as reduced cofactors. So again, these reduced cofactors we can refer to as electron middlemen. So again, going through the citric acid cycle is the way that we fully oxidize our fuel into CO2. But again, oxidation being the loss of electrons, where do those electrons go? Well, those electrons go to FAD and NAD plus and reduce them to become these reduced carriers, and these guys carry their electrons to the electron transport chain, where a proton gradient is generated, and then that proton gradient is dissipated via oxidative phosphorylation to generate ATP. So I know that's a mouthful right there, but we'll get to the details of that as we talk about these different processes. So again, energy in our fuels as electrons is stored in the reduced cofactors here. They deliver their electrons to the electron transport chain, and we generate ATP via oxidative phosphorylation, and those electrons ultimately get delivered to oxygen, reducing it to water. So again, this is how we generate those 2 waste products in our main fuel equation. So I know there's a lot going on this slide in terms of names of things, but there's really only a couple of things that we need to identify, and that's that most of the cellular metabolic processes that we're going to talk about occur either in the cytosol, which is again the fluid part of the cytoplasm, or a specific organelle that's in the cytoplasm called the mitochondria. And so we'll spend a lot of time talking about the structure of the mitochondria in class, but for right now all we really need to know is that the mitochondria is sort of the powerhouse of the cell. So again, lots of other cellular components, but the main ones that we need to worry about are the cytosol, again, the fluid part of the cytoplasm containing lots of the enzymes, and then the mitochondria. So structures for the synthesis of ATP from energy-producing reactions. So once we learn these processes, one of the things that you'll need to know is where do they occur? Are they cytosolic or are they mitochondrial processes? So kind of diverting for a little bit, thinking about how metabolism works, right? Fuels are worth energy because, again, they're a source of electrons, and we can harness those electrons by allowing those fuels to undergo the loss of those electrons, or oxidation. So the concept is that if we squeeze out all that energy all at once, it is we burn a fuel, we physically burn a fuel, We call that combustion, and right, a flaming marshmallow is a good example of combustion. And maybe you may or may not know this, but this is often how calorie content is determined in foods, is they actually physically burn it and they measure the amount of heat going off. And that chemical equation for fuel burning is the same as we see for fuel metabolism. Fuel plus oxygen gives us carbon dioxide, water, and then energy. So when we think about metabolism, what we want to think about is the same chemical equation, except we want to do it under a much more controlled environment so that we can have a harnessing of this energy, and importantly, we want to think about capturing it. So again, if we imagine a fuel as a sponge, right, and it contains energy as electrons, and we think of the water in that sponge as those electrons, what we really want to think about is squeezing out that sponge to be able to harness that energy, harness that water, harness those electrons. But again, having something to catch the water, those are our energy molecules of metabolism. We've already mentioned them. That's that NAD plus and the FAD, which catch those electrons and then become FADH2 and NADH. Again, if we can harness and store that energy in those those reduced molecules, we can ultimately translate the energy of the fuel into ATP. So a little bit of information here on these energy molecules of metabolism. Right? Energy molecules of metabolism have two forms, either high energy or low energy. And high energy ones have the electrons stored as bonds in the molecules, and the low energy don't have electrons. Remembering that electrons represent energy. If they don't have the electrons, they're low energy. And so we're going to talk about different ways that we can have that energy in those electrons harnessed. Sometimes the high energy comes as a function of a high energy bond, as is the case with ATP. Remember when we talked about the structure of ATP? This is a very, very simple way to represent the structure of ATP. We've got these bonds here between these phosphate groups. These are those high energy phosphate bonds. Remember we showed these in our textbook as being squiggle bonds, meaning that they're high energy. So this bond is high energy, this bond is high energy. So again, if we break that bond, we can liberate this energy. So again, hydrolysis of that bond allows us to release the energy, and we can use that to do work in our cells. So that's why ATP is worth energy. We learned that when we learned about the structure of ATP. So again, we can think about high energy bonds like those squiggle bonds. And again, squiggle bonds are found in other forms. When we talked about acetyl-CoA, remember we showed this thioester bond as sort of being a squiggle bond there. So again, here's the high energy bond when we have acetyl-CoA with this 2-carbon acetyl group on the thiol functional group. When we don't have that acetyl group there, we don't have that thioester, so we don't have the squiggle bond. So again, we're going to talk about acetyl-CoA as being kind of the entry point for the citric acid cycle. So once we get rid of that acetyl group, we no longer have a high energy molecule. Another way of thinking about high energy molecules is having reduced forms, right? Because a reduced molecule has electrons. So again, big picture, as fuels are oxidized and they lose electrons, they give them to oxidized forms like NAD plus and FAD. As those become reduced and that they gain electrons or the energy, we're going to then have those harvest them. So here's two examples here. NAD plus is the oxidized form. That's the low energy form because it does not have the electrons. You'll notice here, remember we talked about our B vitamins. So NAD plus is derived from vitamin B3. Once we harness those electrons and we save them as NADH, again, that's our high energy form because we have the electrons. So big picture, we can store energy as high energy bonds and squiggle bonds. We can store them as reduced cofactors here, coenzymes. The one that's derived from vitamin B2 is our FAD and FADH2. We'll talk much more about these in class and some nuances and subtleties that make these different. But ultimately, the low energy form is the oxidized form, not having the electrons. The high energy form is the form that has the electrons. And again, we can see that when we have those electrons, we have to have them, in this case, form bonds with other things. And so we have them form bonds with hydrogen atoms to generate those reduced molecules. All right, so big picture, how do you get food and fuel into your cells, right? So we first have to start with digestion, right? Digestion, as we learned about, is a hydrolysis reaction. We need to break those either glycolytic or peptide linkages or those ester linkages in our lipids, and we generate monomeric forms of these fuels so that we can get them out of the digestive tract and into the bloodstream. So again, proteins must be hydrolyzed to amino acids, carbohydrates hydrolyzed to monosaccharides, and triglycerides as lipids must be mobilized as free fatty acids. And again, we need to think about how these molecules are able to travel in the bloodstream then, right? Because we've got solubility issues. We really don't have a problem with carbohydrates or proteins as their monomeric forms because monosaccharides and amino acids are very water soluble. However, lipids are not very soluble, and triglycerides, or even for that matter free fatty acids, are packaged with other lipids like cholesterol, and they're put into particles called chylomicrons. And we'll look at the structures of those much more closely in class. But again, you can think about these as being large vesicles, if you will, vessels that are very hydrophilic on the outside because they're kind of made of some protein on the outside, but then they house the hydrophobic lipids on the inside. So we're going to take a little bit of time here and just talk. We've already talked about the details of these structures very intensely in class in previous chapters, but we're just going to review them and remember how we take these polymers and break them down into their monomers. So for carbohydrates, the main carbohydrates we're going to see might be some disaccharides like sucrose or lactose, but the starches that we consume as polymers of glucose, starch digestion actually begins in the mouth with salivary amylase. And again, we begin breaking those glycosidic linkages down to get to smaller and smaller pieces. Again, free glucose is going to be able to be immediately absorbed into the bloodstream. But following digestion in the stomach, further processing of these smaller saccharides in the small intestine with some additional enzymes here that allow us to fully hydrolyze these glycosidic linkages, getting down to these monomeric sugars like glucose, galactose, and fructose and mannose to a lesser extent, and those again can be taken up into the bloodstream. So again, complete digestion requires a full transit through the small intestine. And again, as we've seen in our membrane transport chapter, that small intestine and those intestinal villi, that's where most of the absorption of our fuels take place. So remember, anything that's not digested is not going to be absorbed. So something like cellulose, right? We don't have enzymes that can break that beta-1,4 linkage in that glucose polymer, and so it's not digested in our intestinal tract, and so we aren't able to take that glucose and use it as a fuel. But remember that fiber is very important in the health of our digestive tract as it allows materials to continue to move through the digestive tract. All right, so lipids, right? So again, here's, when we had our little upside down W to remember the structure of triglycerides, we've got fatty acids that are esterified onto a glycerol backbone. So lipases in the small intestine are responsible for hydrolyzing those ester linkages, liberating free fatty acids, and those free fatty acids are able to pass out of the intestinal tract into these intestinal cells, and within those cells we're able to then facilitate packaging into chylomicrons, again complexing with proteins to make these protein lipid particles that are much more water soluble. So these chylomicrons are what are going to circulate through the bloodstream, and their first stop is often going to be the liver, where then they're going to be able to deliver their contents. So we'll get into a good bit more of that in some other chapters here. But again, we have unique issues with dietary fats being nonpolar, so we secrete bile and store it in the gallbladder. And again, that is cyst digestion by allowing us to emulsify those fats, making mice cells that are much more susceptible to these pancreatic lipases doing their chemistry. All right, so again, we are going to generate these chylomicrons. You can kind of think of chylomicrons as giant transport vehicles that allow us to be hydrophilic on the outside with the protein component and hydrophobic on the inside with all of the lipids that are being carried. And so again, we're going to transport these through the bloodstream. Proteins, right? Protein digestion begins in the stomach, where we denature proteins. Again, once we get to the small intestine, we're going to have our series of our serine proteases, right? We've got chymotrypsin, trypsin, elastase is another one that we learned about. Pepsin is another intestinal protease that we can use, hydrolyzing those peptide bonds, generating free amino acids that can then be uptaken into the bloodstream and distributed to tissues. So very interestingly, amino acids are often one of the first fuels that can be used following digestion because they immediately get into the digestive tract. So we'll talk about that a lot more in class. All right, so now we've got all of our fuels into the bloodstream. So now what do we do? So first step is we're going to talk about glycolysis. And glycolysis is, again, a specific pathway to glucose catabolism. But because glucose is a primary fuel in your body, it's an important pathway to study. Your body runs on glucose, and so it's really important to understand how this works. Importantly, this pathway is what we call anaerobic. So this is one of the only pathways by which you can generate energy. You can generate ATP in the absence of oxygen. So there's times metabolically when this operates very easily within your cells. There's certain organisms that operate exclusively by anaerobic glycolysis. When we do our beer fermentation, we'll talk about the glycolytic pathway in much more detail. But so here's some bullet points that I kind of want you to remember about glycolysis. 10 reactions, so remember the number 10. Breaks down glucose, and it's a cytosolic process. So we want to remember where it occurs and the number of reactions that it occurs with. Again, an anaerobic process, so it does not require oxygen. And I want you to remember that it makes energy quickly, but not efficiently. And the reason it's not efficient is we're left over with pyruvate at the end. That's still a fuel that has a lot of water, if you will, to squeeze out of that sponge, but we're not able to get that energy out. So it's an anaerobic process, does not require energy, makes energy very quickly, but it's not very efficient because we're left with a fuel molecule that under anaerobic conditions we really can't do anything more with. So at the end of glycolysis, I want you to remember that a 6-carbon glucose molecule will give 2 3-carbon pyruvates. You'll get 2 ATPs, again, which is the desired immediately used cellular currency, and then we'll get NADH, which is an alternative currency. So that's the nuts and bolts I want you to remember about glycolysis. We're going to look in a little bit more detail right now on the glycolytic pathway, and the last chapter we'll study in Biochem 1 here is actually the entirety of the glycolytic pathway, looking in much more detail as this. So in this very simple schematic here, what I want you to look at and remember is that each one of these little spheres, these blue spheres, represents a glucose molecule. And again, little p's that are shown in yellow here are phosphates. So we can think about glycolysis proceeding in 2 stages. In the first stage, which we have shown in purple here, we call this the energy investment stage because we actually have to use up ATP to get this process started. And the whole idea is that we're going to be decorating our glucose molecule here with phosphates, and that creates high energy species. So we're going to generate high energy species in this first phase, so this is an energy investment phase. And then in the second half of the pathway, this is the second stage shown in green here, is our energy generation. Once we've made these high energy intermediates, these high energy phosphorylated intermediates, we can then use them to synthesize ATP. And it's not written on this slide, and this is something that we'll talk about in much more detail in class. This process of synthesizing ATP is called substrate level phosphorylation. So that might be something you want to write down because it's not part of these slides. Substrate level phosphorylation is basically using a phosphorylated intermediate that's higher in energy than ATP, and so we can run this ATP hydrolysis reaction in reverse. So again, hydrolysis would take ATP to make ADP. If we have something that's higher in energy, we can actually thermodynamically couple these reactions, and we can run the synthesis of ATP from ADP. We can run that reaction in the forward direction by using the energy of these high energy phosphorylated intermediates. And we do that at 2 different stages here in glycolysis. This molecule called 1,3-bisphosphoglycerate or 1,3-BPG is higher in energy than ATP, and so is phosphenylpyruvate. When we study glycolysis, we'll learn a lot more about why that is. But again, big picture, glycolysis is 10 reactions, it's cytosolic, it's anaerobic, it makes energy quickly but inefficiently, and it occurs in 2 stages. The first stage is energy investment, where we're decorating our glucose with phosphates, and then the second stage allows us to use those high energy phosphorylated intermediates to drive the synthesis of ATP in a process called substrate level phosphorylation. So herein is part of the problem of glycolysis. For glycolysis to continue, you can see here in step 6, we need NAD+. That's a necessary co-substrate that we need for this process. So if we've run out of NAD+ in our body, we can't continue to have glycolysis occur. So there's a couple of ways that we can remedy this, and how we remedy this depends on whether or not there's oxygen around. Under anaerobic conditions, we're going to kind of operate in a unique way. So under anaerobic conditions, we're going to take this pyruvate molecule and we're going to reduce it to lactate. And again, this being a reduction reaction, pyruvate to lactate is a reduction reaction. Remember how we'd identify that that's a reduction reaction. We can see the chemistry is going on at this carbon atom. We go from a more oxidized carbonyl-containing functional group in our ketone to a more reduced alcohol. So again, this being reduction means that we must couple it with an oxidation. Well, the oxidation is the regeneration of NAD+ by the oxidation of NADH. So again, under anaerobic conditions, that's what's going to happen. This is actually what happens under, for example, sprinting or weightlifting or very short-term exercise strategies where your muscles will operate anaerobically. You can do this for short periods of time, so it's a quick fix for fast production of energy, but it's inefficient and it's not sustainable. So again, you can really only maintain this activity in your body for about 4 minutes. Under aerobic conditions, a very different scenario is going to happen. Under aerobic conditions, what we're going to happen is pyruvate is going to be committed to chemistry that's catalyzed by pyruvate dehydrogenase. That enzyme name is not listed here, but it's an important step that snips off one carbon dioxide and takes the other 2 carbon atoms and appends them to our CoA substrate to give us acetyl-CoA. And hopefully you remember that this thioester bond is a high energy intermediate. So we've saved the energy of this molecule by generating this high energy intermediate. And it's important to note that once you make acetyl-CoA, you are kind of committing this to further aerobic oxidation in the citric acid cycle. So again, this process happens in the mitochondria. So we're going to have to transfer our substrate to a different compartment in the cell. So we're going to have to go across the mitochondrial membrane and get into the mitochondria. And again, this process starts to generate some of our CO2. We only commit to this process if we have oxygen available. And as we'll see here, it is a way that we regenerate, we're able to regenerate our NAD plus. And so we're going to have that be an important fate that we'll see here. All right, one other possibility, we're not going to see this at least in your body, but we will see this in the laboratory. One other fate for pyruvate is that pyruvate can undergo, again, a reduction, this time not to lactate, but to ethanol, and we can see the concomitant oxidation of NADH to NAD+. This generates ethanol and liberates CO2. And these 2 products are really important because in our beer production, or wine production for that matter, that's how it becomes alcoholic, and this is how it becomes carbonated. So we'll talk a lot more about the details of that process in lab as we begin our beer production. So a couple of other things I want you to remember about glycolysis. We'll get to the details of this when we study glycolysis in chapter 15. For all of our metabolic pathways that we'll study, we'll learn about different spots that are kind of gatekeepers or regulatory steps. So I want you to remember the number 3 because that is the rate controlling enzyme of glycolysis. So the 3rd step, and we're going to learn about this enzyme, it's called PFK, it's phosphofructokinase. It's responsible for regulating flux through the pathway. So it's regulating how much fuel we are allowing to go through glycolysis. And herein is part of the reason why we think fructose is so important in the obesity epidemic nowadays, is when we think about fructose metabolism, and we'll talk about this later on in the course, fructose metabolism in the liver is not regulated because fructose actually enters in much further down in this metabolic pathway, so it bypasses the regulatory step. And in doing so, it kind of doesn't allow proper control of this pathway. And what happens is we have a lot of unnecessary conversion of carbohydrates to lipids and to fat. And so it's is an important contribution, we believe, to obesity. And again, this is a very large infographic. I'm not going to really go through much of it. We'll actually spend some time in class talking about this. But when we think about fructose metabolism, because of how fructose is metabolism or how it's metabolized, and because we tend to see much more fructose in our diet than we ever had when we just consumed natural foods, we kind of overwhelm this regulatory system in our body, and it really is thought to contribute significantly to the obesity epidemic. So we'll spend more time in class sort of talking about that. One other thing I kind of want to think about is we will spend a lot of time kind of talking about blood sugar regulation. Not only is diabetes going to become a worldwide health epidemic, it's just important to think about how your body regulates its own blood sugar. So again, numbers to kind of think about and remember is blood sugar must be maintained at less than 100 milligrams per deciliter if you're fasting, or less than 140 milligrams per deciliter if it's within 2 hours after eating. So when they do and monitor blood sugar levels, that's kind of the numbers that we're looking for. Remember, we learned about blood sugar as 5 millimolar. Again, about the same values that we have here. So again, if you are not diabetic, if you haven't eaten and your blood sugar begins to drop, what are ways that you can fix this? So some of the terminology that we'll use to talk about this is top down and bottom up. So again, top down is kind of catabolic processes that are going to generate glucose as an end product. So again, catabolic meaning we're breaking down other things. So from an exogenous source, exogenous again meaning external, we can eat and break down starches, and breaking down those starches are going to give us the glucose that we need. However, if we're not going to eat, right, we can have endogenous sources of glucose. So we've already talked about glycogen metabolism, so that's a way to take an internal source of animal starch and break it down and give you the glucose that we need. So we refer to that as top down. We mentioned already in class this bottom-up approach, and that's the idea of using an anabolic pathway, so synthesizing glucose from smaller precursors. So again, we can make glucose from proteins, from other non-carbohydrate molecules by a process called gluconeogenesis. And we'll talk about gluconeogenesis in our carbohydrate chapter on glycolysis, because gluconeogenesis is really the reversal of the glycolytic pathway. All right, we're going to go a little bit more quickly through kind of the next two processes here, remembering that any fuel ultimately will get funneled down to acetyl-CoA and then can be part of the citric acid cycle and beyond. The citric acid cycle is also referred to as the Krebs cycle, and again, it's the pathway by which we catabolize acetyl-CoA, and again, that's the main metabolic breakdown product for any fuel. So carbohydrates, fats, and proteins can all be made into acetyl-CoA. So some numbers that I want you to remember for the citric acid cycle: it's 8 reactions. It occurs in the mitochondria. It's aerobic, so that it will not occur without sufficient oxygen present. So even though we actually don't see oxygen present in this part of the metabolic pathway, we will not commit fuels to the citric acid cycle unless there is sufficient oxygen around. So that's an interesting distinction, that it is an aerobic process in that it requires oxygen but doesn't use it directly. So you might actually hear that some people say that the process itself is anaerobic, but it does not occur under anaerobic conditions. It requires aerobic conditions. We do not commit fuels to the citric acid cycle unless we have sufficient oxygen to handle all of the reduced cofactors that are produced. So again, it is a catabolic pathway, so we're breaking things down. We're breaking down acetyl-CoA, and the 2 carbons in that acetyl-CoA become carbon dioxide. So again, there's no more carbon left in our fuel at this point. We generate 1 molecule of GTP, which is actually energetically equivalent to ATP. And then we generate a significant amount of alternative currency, so 3 NADHs and 1 FADH2. And so now the problem is that most of our energy is tied up in these middlemen, so we need to think about what we do with them. Again, just looking at a little bit more detail in the citric acid cycle, looking at these little spheres. Again, these represent carbon atoms that we're going to have here. And so each of these carbon atoms is going to undergo different kinds of chemistry in the citric acid cycle. And so if we want to, we can think about the cycle in terms of two halves, kind of a stage one that's kind of the first half of the clock and a stage two, which is the second half of the clock. And ultimately what happens is we mix two substrates together to generate a 6-carbon piece. We snip off 2 carbon atoms in the first half of the cycle, and then the second half of the cycle is really reconverting this 4-carbon piece into this 4-carbon piece. So we'll get to the details of that, actually not until Biochem 2, but that's sufficient for you to understand kind of what happens. 8 steps. The first few steps are liberating CO2. The next steps are regenerating the necessary metabolite that we need to continue this cyclic process. I'm realizing here as I'm pointing to these spheres, I think when we had, I'm going to flip back just for a second, when we had our picture here of glycolysis, these are carbon atoms. I think I maybe mistakenly said that these were glucose molecules. This is a glucose molecule made up of 6 carbon atoms. So I just want to make sure to correct that there. All right, so again, taking stock of what we've done so far is we have fully oxidized a 6-carbon glucose molecule. We've liberated that carbon in the fuel as 6 CO2 molecules. We've generated 2 molecules of ATP directly by glycolysis, 2 molecules of ATP directly by the citric acid cycle, and then we have lots of reduced cofactors here that we're going to take onto the electron transport chain. So again, where's all the energy in our fully oxidized fuel? For ATP is not very much. We've got it tied up in our alternative currencies. So we're going to squeeze the rest of the energy out of those alternative currencies in the process of electron transport and oxidative phosphorylation. So again, electron transport and oxidative phosphorylation, which I frequently just abbreviate ETOP, is a process that is mitochondrial, and electrons are going to flow through a series of complexes, and they're going to generate a proton gradient. And so remember, we talked about gradients in chapter 10 when we talked about the energy that is harnessed in a gradient. And so we can use that gradient to generate ATP. So again, the electrons are flowing through a series of complexes. They're ultimately delivered to oxygen. Delivery of electrons to oxygen means that oxygen's reduced, so it makes water. And then again, oxidative phosphorylation is the process by which the energy from that proton gradient is used to make ATP. So just to kind of draw a parallel here, remember in glycolysis we talked about making ATP by a process called substrate level phosphorylation. That's a process wherein we use a molecule that's higher in energy and phosphorylated, and we use that. It transfers its phosphate to ADP to make ATP. In this situation, we actually are going to use the energy of a proton gradient to drive the synthesis of ATP. We're also going to make ATP from ADP, but we're going to use the energy of the proton gradient to provide that energy. So again, here's a very simple cartoon representation to kind of show what happens here. We've got a bunch of different complexes. You can see arrows here showing that we're pumping protons here as our electrons kind of transit through. And then this complex, complex 5 over here, we have protons traveling in the other direction. So this is used to dissipate the proton gradient. When we dissipate that proton gradient, we can use that energy to run the ATP hydrolysis reaction in reverse and make ATP from ADP and inorganic phosphate. So again, we can see here when the electrons finally exit the electron transport chain, they're going to be delivered to molecular oxygen, and that's going to generate water. And importantly, in this process, when we commit these reduced cofactors to the electron transport chain, we regenerate these middlemen in their oxidized form so they can return back and do some more of their chemistry. And we'll learn the details for how that happens next semester. So a couple of just little interesting side notes that we want to talk about. Some of these we've mentioned already. So again, this kind of represents the same process that we saw on the previous slide. We've got proton gradients being generated by electron transport in those different complexes. We've got ATP synthesis occurring by dissipation of that proton gradient, but this blue protein over here represents what we call an uncoupling protein. So remember, if we couple these two processes together, all of this proton gradient is going to be reflective of the quantity of fuel that went through fuel oxidation and metabolism. So normally these two processes are coupled. The extent of the proton gradient is directly proportional to the amount of fuel and thus directly proportional to the ATP that we produce. If we have another option for proton translocation to happen, we can actually uncouple this process, and we can use this proton gradient not to make ATP, but to generate heat. And this serves an important biological process, and that we can operate this process to do what we call thermogenesis, or the generation of body heat. If you're cold and you shiver, that's what we call shivering thermogenesis. The physical movement of your muscles, generates heat. But for hibernating animals and for infants, for example, they need to be able to generate heat internally without movement of muscle. And so this is generally how this happens, is we have a lot of these uncoupling proteins in brown fat, which is very rich in mitochondria that have these uncoupling proteins. And so they will metabolize their fuels, largely lipids, and rather than synthesizing ATP, which they don't need because they're not doing a lot of movement or other activity, they'll use that proton gradient to generate heat. So again, a really important natural biological process, biochemical process. But one of the things that we've already talked about is the fact that we can have drugs like 2,4-dinitrophenol that actually will uncouple this process. So rather than using an uncoupling protein that will allow us to do this, remember we learned about 2,4-dinitrophenol in our membrane transport chapter. This guy, having a pKa that's near neutrality, can effectively dissipate a proton gradient, because once you pick up a proton, you become neutral, you can travel easily through the lipid bilayer, and you can dissipate a proton gradient in this way. So in the 1940s, this became a very popular diet pill. And we've already talked about this a few times, I think, in class now. The problem is that sort of a dissipation does the same type of generation of heat as this one does. And so unfortunately, when people consumed too much 2,4-dinitrophenol, it caused extreme elevations in temperatures and these fatal fevers. So that's kind of the overview in terms of the slides that I have for metabolism. I just want to show you one last thing. I'm going to show you a little video that we have here that is available. If you just kind of go to YouTube and Google cellular respiration, you'll find this one. So I'm going to play it. I may stop it from time to time to just kind of refer back to different things that we have talked about in this lecture thus far.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins.
So you can kind of see there, they were showing the small intestine. And what they're going to show here is we're going to focus on carbohydrates in this animation, but they're going to show carbohydrates coming out of the intestinal tract. They're going to go through these intestinal cells, and they're going to end up in the bloodstream.
The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells.
So again, you can see here that we're going to see all of these black spheres represent the 6 carbon atoms of glucose. And remember, glucose being fully oxidized will generate CO2, H2O, and importantly, ATP.
Cellular respiration is a 4-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of 2 distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, 2 ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the 2 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants.
So again, that was glycolysis. There's a lot that was going on there. Go back and watch that a few times and see that you can see where electrons are being harnessed as our reduced cofactors NADH. And you can see the different points at which we're adding and decorating our glucose molecules with phosphate, and then we're using those high energy molecules to make ATP by substrate level phosphorylation.
In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria.
So the next step that we're going to talk about, because they talked about it occurring in 4 steps. So the first step is glycolysis. The second step they're going to talk about is pyruvate dehydrogenase. Then they're going to go into the citric acid cycle, and then to electron transport and oxidative phosphorylation. The rest of these processes, as they're mentioning here, occur in the mitochondria.
And the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA.
And there again, you can see the 2 CO2s that have come off there. We're also generating some NADH in the process.
In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water.
So just to highlight, the first part that they just showed there, that was the snipping off phase, where we make some energy and we get rid of 2 carbons of CO2, and now we're left with this 4 carbon piece that we need to remake into that oxaloacetate that they talked about.
Pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule.
So again, that was the kind of stoichiometry that we had. All 6 carbons from glucose are made into 2, 4, and these were from pyruvate dehydrogenase, 6 CO2s, and then we can see all the NADHs and FADH2s that are produced. Just a little bit of ATP at this point, so we need to figure out how to squeeze the rest of the energy out of these reduced cofactors.
In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane.
So again, here's all of those protons that are being pumped in this direction. And so what we'll see afterwards is that proton gradient is released back through here, and this is used to make ATP.
When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain.
So I encourage you guys to go back and watch this a couple of times and watch this guy over here. You'll see molecular oxygen come in and then it actually gets split. We add protons and we add electrons, and that's how we make water.
All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell.
And I'll make sure to let you guys know there's different numbers. We're going to learn a number that's 32. We'll talk about why it's different from what we see here. But again, 32 to 38, and it really depends on the individual. Different people have different kind of amounts of uncoupling proteins and so forth that can affect the amount of ATP produced from a single glucose.
In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
All right. So again, that is basically the summary, the overview, if you will, of cellular respiration via glycolysis, pyruvate dehydrogenase, citric acid cycle or the Krebs cycle, and then electron transport. So I encourage you, maybe watch that little 6-minute video a couple of times. You can probably find it easily on YouTube yourself. But I want you guys to get to the point where, not that you rewatch this entire lecture because it's lengthy, but that you are able to kind of think about and have this terminology be familiar for you. And that's it.